Thanks, Nate. Yeah, so I'm really happy that I get to talk to you about EM, in particular about the beaming of worms. So let's beam up some worms. So here's a worm. There's some weird stuff on it. I'm not sure what it is. Maybe it's just a flake of my skin. It doesn't really matter. The fact is, we see the worm. So this is scanning electron microscopy here. And this is a dower in this case. You notice it's way smaller than an adult would be. And then we deposit some platinum to ground the worm. It gets a nice little belt of platinum. And then we beam it. There you go. So the worm is beamed away. And this is sped up about 10 times. So it's not quite as fast as Star Trek, but it's gone. Now the question is, where did it go, right? Where did I beam it to? So just before and after. So where is it now? I did not beam it to another planet. No, we can't really do that yet. But I sort of beamed it into my computer. Here it is. So now, kind of like in Star Trek, I guess, by scanning it, by beaming it away, we also scanned it, destroyed it in the process, and we reassemble it, not on a different planet, but in silico. And with this technique, with this worm beaming, we get this beautiful data set that I can look at for like hours, where it's perfectly smooth, right? There's no cutting involved, so it's all smooth and no folds, no dirt, and you can look at it from any angle. It's beautiful. Oh, right now we're going into the nerve ring. There we go. This is the nerve ring. Isn't it cool? All right. Now, of course, let's back up and I'll show you how I got there, right? So, I mean, first of all, we do all this beaming mostly to just get access to the interior of the worm. Well, and if we have access to that, we can just use regular scanning electron microscopy to scan this surface to get an image. And basically, this technique, which is called FIB-SEM, focused ion beam SEM, works just by alternating milling and imaging. So once we have the initial access, we just scan, get an image, then we use the FIB to remove a little bit of material, in this case about 8 nanometers, but you can do even less. And then you scan again, and you remove a little bit of material, and you scan again, and then bit by bit, you create your stack of images. Yes, right? Here, then you see it. Then speeding it up a lot, just looks like the worm is vanishing. All right, now, how do we get there? We start with the worm. So this is a dower. And then we do high pressure freezing, as Mia explained. So we put it in the carrier, we put it in the machine. This is a high pressure freezing machine. We fill it up with liquid nitrogen. We put the worm in. We close the lid. An awful lot of noise that you think, oh, now the machine is broken, but no, that's normal. Now the worm is frozen. We take it out. It's now in a bubbling liquid nitrogen. Here are the plastic pieces with the golden carriers that the worms are inside. And we transfer it. Then we do the freeze substitution, just as Mia explained. And then in the end, when we free the worm from the pellet, we have a single worm that's now black from the heavy metal staining. Now, so far, this has all been just standard procedure, right? Like we always do for worms. But now comes a little bit tricky part, because I cannot put this worm now in a block of resin, because how would I find it in my SEM? I would just see a plastic surface, but where's the worm? I need to see where I want to beam, right? So this is why we established so-called minimal resin embedding. This is really key, so we can find the worm. Funnily, like when we're thinking about it, we need to somehow make that we can see the worm after the embedding. How do we do this? We came up with all kinds of weird contraption, how to make like the resin flow off and stuff. And in the end, the by far simplest method was best. It's so simple, in fact, that it was almost embarrassing to publish it. Where you just, where's my mouse? Here's there's a drop of resin, and you just take like a needle or a toothpick or something, and you push the worm out of that. You push the worm out of that drop until it kind of seems to be dry, right? So it's across the plastic surface until it seems dry, and that's it. Now there's no resin, like there's only a very thin layer of resin outside of the worm, but it's still infiltrated. So inside of the worm, there's resin. So this might be the world's tiniest block. And the great thing is now that worm is pretty much naked, we can also evaluate, like, look at this, we can even see the cuticle ridges and everything. But this is even a good method if you want to just do SEM and just look at the anatomical features of the worm from the outside. We can even see the amphid channel here on the last picture. Yes, and then we FIB. With this machine, like this is a FIB-SEM, looks pretty much just like a regular SEM with some added stuff, right, to create the focused ion beam and the platinum deposition. Then we have a sample in there. We kind of bring it in position. We look for the worm. Great, we found it. And then we can start the milling, the fibbing, the beaming, whatever you want to call it. And then we get such amazing data sets. And because we can, like, we don't have to section, we beam, and we only remove a few nanometers, this is actually an isotropic data set. So that means, this is why we can also look at it from every angle. It's isotropic, meaning we have the same resolution in X, Y, and Z. In this case, it's about 8 nanometers. Maybe you can even get down to 5 nanometer voxel size. And yes, this is the data set that we got this way. And yeah, let me show you some, like, this is a nice video. Robert Kiemse was at the company Zeiss at the time, liked our data set so much that he made this promotional video, I guess, to promote the machine. But let me show you what you can see, what you can expect with such a FIB-SEM data set. So here from the ventral nerve cord, we can see vesicles, right? You see synaptic vesicles here. We cannot resolve individual ones, but we see that they're there. We see the dense projection, so we can make out that there's a synapse. We also see, like, I don't know, all kinds of stuff, mitochondria, muscles, lipid droplet. This is how it looks like. And over here, there you have a Golgi apparatus. So we also see, like, organelles and stuff. All right. And here, we're more in another region, in the head part. Again, nucleus, this is from the H cell, here in D, another Golgi apparatus, another synapse. And here you see some ER inside of a soma. So yeah, you can see a lot. It's a very nice data set. Here at the top part of the worm, here, you can even see microtubules. Here, OLQ is a neuron that has a dendrite, so that means it has microtubules. Here, the ring of 9 microtubules. Oh, sorry. Let me go back. So you cannot really resolve them really well, but you see that they're there, right? So it's at the edge. Many things, I admit, are a little bit at the edge, but you can at least see that they're there. And yes, here are just some impressions, what you can see: muscle, AFD neuron, the sheath around, like this black stuff around is the sheath around the sensory neurons, and so on. All right. Now, this is, well, you can do a lot with such a data set. Really, you can use it. We give it to you, right? You can use it for whatever analysis you want. What we now published is a comprehensive analysis of the interior sensilla region. So we pretty much segmented everything that's somehow involved in sensing something. And really, the worm has like a whole laboratory at the nose tip, right? And so all these different neurons, this is now the first really comprehensive reconstruction of these sensilla in the Dauer. And so now, yeah, we have a volumetric reconstructions of all the interior cell endings in the Dauer. Right, and this is the full data set. This was 10,000 individual images. It took about 2 weeks to acquire that. And that is 2 weeks of the FIB-SEM machine running 24/7. So since we only remove a little bit of material at a time, it really takes a long time. And yeah, in this data set, we also include the nerve ring. So this is why it can only be used for connectomics analysis. I've shown you, right? You see the synapses, so you can look at the wiring as well. And we also did a little bit of that. Okay. Now, after all these, what's the word? Like, after singing the praises of FIB-SEM, now, of course, let me get to the downsides. Well, the first one is that, honestly, it's not very accessible. I mean, who has a FIB-SEM? It's pretty expensive, but I mean, you know, EM people are very friendly and they love to collaborate. So if you want to do FIB-SEM, why not just go to your local imaging core facility or EM facility and ask them for help? And if your facility doesn't have one, well, for example, contact me. I'll get you in touch with someone who does. I promise we'll find someone for you. And then you can get access. Yes. Another downside, though, is that the ROIs can be hard to find, right? This is the whole point why we had to do this minimal resin embedding. And even then, I mean, now you see the worm perfectly from the outside, but of course, it's still kind of tough to target something that's on the inside, right? Because you first need to kind of fib it away to get access. So once you see it, you already destroyed it a little bit, right? So you kind of have to start imaging before your region of interest comes into view. Because if it's in view, it has already been destroyed, right? So that can be a little bit challenging maybe sometimes. And then maybe you have to like sit there for a long time waiting for your structure to appear to actually acquire the full data set. Right. Then, honestly, the volumes are relatively small, right? I mean, I told you for this, like, only the head part, the first fourth of the Dawa larva, which is a very small worm, right? Took 2 weeks to acquire 24/7. So doing the full worm would be like months, and doing an adult worm, yeah, I mean, then the machine would have to run for many months. So typically people do not more like subcellular structures, or maybe like a single cell, or like part of a dower. It's feasible to do a full dower or larval stage, or even an adult, but be aware of that. It's not really for big volumes. Then you need some of the other techniques we've seen, like array tomography, or maybe serial block phase or something where you cut the sections. Right. And finally, as I've shown you, the resolution is limited. I mean, the resolution in Z is unparalleled, right? I mean, as Ben showed you, with a very good microtome, maybe you can cut down to 30 nanometers, but we can easily go down to 5. So in Z, it's unparalleled, but in X, Y, it's also just 5, right? So as I've shown you, you cannot reliably resolve synaptic vesicles, for example. But if this is enough for you, then FIB-SEM might be a great way to acquire volumes. All right. Now, here's some acknowledgements. I particularly want to highlight Sebastian Markert, who did really most of the work of the data analysis here. Sadly, for us, he has now left science, and this is why he now has to dress much nicer. And he had a lot of help from Daniel Whitfleet from May Sense Lab, and also from Ben here. And yeah, I'm pretty sure we will, like, this data set has so much, we will still work on it in the future. Okay, there's some logos. Yes, and that's it. Thank you very much.